Good afternoon, everyone. We'll get started with the afternoon program, which will start with a state-of-the-art lecture by the indomitable Dr. Tim Gretton. So it's my pleasure and honor to introduce Dr. Gretton. He is the Deputy Branch Chief and Senior Investigator at the NIH Thoracic and GI Malignancies branch at the NCI. He's also co-director of the NCI CCR Liver Cancer Program. He completed his medical medical school at the Christian Albert University in Germany in 1986. He then went on to obtain his PhD in Austria, and then subsequently came to the United States, where he completed a postdoctoral fellowship in tumor immunology at Johns Hopkins in 1998, and then completed medical training in internal medicine, hematology oncology, and gastroenterology hepatology at the Hanover Medical School. After that, he went on to faculty at Hanover prior to coming to the NIH in 2010, where he has been since that time. So in a nutshell, Dr. Gretton has been at the forefront of his field. He has been studying tumor immunology since the 90s, well before it became a topical area of investigation. His cutting-edge science has resulted in a very impressive publication track record with over 250 peer-reviewed papers in incredibly high-impact journals, including Science, Nature, Cancer Discovery, New England Journal of Medicine, and Cancer Cell. He is world-renowned for his work and has served on the editorial board of essentially every major GI journal. He's also been the recipient of multiple awards, including the NCI Director's Innovation Award, the NCI Director's Merit Award, and more recently in 2023, he was awarded the NCI and CCR Director's Award for Outstanding Mentorship. He is co-chair of the Center of Excellence in Immunology and co-director of the NCI CCR Liver Cancer Program. He has a particular interest in the interactions between immune cells, immunosuppressive cell types, and the microbiome, and he will be giving the state-of-the-art lecture today entitled From Mice to Men and Back to Mice, Basic and Translational Research Studies in Cholangiocarcinoma. Thank you so much, Tim, for being here with us.
Thank you, Samira. Now I know why I got 60 minutes, because your introduction took so long. Thank you so much. I don't know what to say anymore, but so let's get started here. I will only talk about immunotherapy, so I know there's a lot of interest in targeted therapies. I don't know that well, so the majority of the talk is immunotherapy, and as you all know, immunotherapy is playing a big role in biliary cancer. Basically, now it has become standard of care treatment. You know these studies, these study results. Have you really looked at the study results? Overall survival benefit of 40 days or 55 days. I think, that speaks for itself. It tells us that the treatment is working. But it also tells us that there is way more to understand and to improve, and that we need to improve. And that we basically have to take advances in basic science to provide new opportunities to improve the understanding of immunological mechanisms, and that's, kind of what we're trying to do in our group. And the focus of this talk is actually is something, or our work is, that we have to keep in mind that, we're treating patients. I'm an MD, and I know that MD can stand for mouse doctor, but we're doing this primarily, to advance treatment and treatment options for patients. And the theme of this talk will really be, bouncing back between patients and preclinical studies. So let me give you an outline what I thought I should talk about. I thought I'm going to summarize some of it, what you already heard, some of it this morning, where we talk about the tools, how do we do these studies, what can we actually utilize. I will tell you a little bit about true studies that are really done only in cholangiocarcinoma, where we study the immunology and the immunotherapy. And then the majority of this talk is indeed on clinical trials specific treatments depending on mutations, correlative studies, and then this ping pong between correlative studies and functional mechanisms. And I think, we have some very interesting data, and this is the majority of the talk is actually I've never shown before, so I think it's going to be interesting. And then last but not least, I couldn't resist. I'm going to talk about something that's really cool, so stay to the end. It's microbiome, gut, and the brain. So, we usually, those people who, know me, we're trying to push things and think about maybe new approaches that we can do, and we have some, I think, very interesting findings there. So let's get started with the tools. So, you heard it this morning, how do we do this? If we have preclinical models, obviously, we start out with patients, we have tumor resected and or biopsies, and we can either use them directly for sequencing studies, but if we want to do functional studies, there's different ways, obviously, of culturing them, developing cell lines, PDX, going into GEM models. The important thing when we talk about murine models is that there's so many factors to consider. The mouse model will never be the same that what we have in a patient. And we really have to adopt and choose the right model for the right question. We have to consider location. We have to see that these tumors express the right markers. The question of precancerous lesion that we're discussing about this morning. Obviously, the molecular profiling plays a role. The questions of response to therapy. Does this tumor really look like what we see in a pathological specimen? And are there potentially targetable mutations similar to what we find in patients? And there are various models available. These models can either be derived from patient samples that we put into mice cell lines, or we can induce it in mice directly using by genetically engineering or chemicals. And there is one advantage, a technical advantage, that I just want to bring to the people to attention that don't do this every day. Because of the anatomy of the liver, there is a technique which is called hydrodynamic tail vein injection, which is widely used by investigators. And basically what you can do is you can use a plasmid of interest, and you can inject this plasmid through the tail vein into the mouse. The trick that you do is you take a relatively large volume, or I should say a very large volume. So this is actually 2 milliliters that you inject into the mouse in 7 seconds. Now, I'm just going to tell you, a mouse actually only has about 1.5 milliliter blood. So you basically double this volume. So what happens is you induce transient right heart failure in this mouse. So there's stasis before the right heart, which is in the liver. And so you have a high concentration of plasmids, and these plasmids can actually stay there, and they can transform hepatocytes or transduce hepatocytes. And this is how a lot of these models work. I have listed some of them, and we have heard about them. There are different genetic alterations. Some of them, the majority of the models that I have listed here are done using these hydrodynamic injection techniques. Some are the genetic models. You heard the extrahepatic models we heard from Samira that talks about the models that she has developed. And I think they all have their certain advantage and disadvantage, and it's important to remember that you basically use really the correct model for your question. Okay, so once we have these tumors, what are we going to do? Obviously, we can measure tumors, and that's straightforward, but I thought, I share with you some of the more modern or more interesting things. We can do actually imaging of these tumors. So this is an example where we perform in vivo imaging. So if we want to activate T cells and we want to see how the T cells go into the tumor, we can actually see this using different systems where we label tissue and antibodies with different fluorochrome, and then open the mouse and have a camera there, and then you can actually look right into the liver and into the tumor. And in this case, you see specific CD8 cells how they actually, migrate through the tumors. And this is helpful because it tells you something about, different aspects, where they go, how they go there, and how they're potentially affected by the metabolism, for instance, or, the environment. Similar to human, we can also do imaging of tumors. And this is, again, can be very helpful if we do our study. So this is actually a conventional CT scan of a mouse. So for the medical doctors in the audience, you will recognize we put the mouse, unfortunately, the wrong way around, because actually the back is up here. We are used to see this down here, but this is just to show you. So this is the spine of the mouse. This is the ventral surface. And what we combine this with is actually contrast dye that accumulates in macrophages. That's why this area of the liver here is white and the tumor appears black, and we can see vessels, gallbladder, etc. And now if you actually scan the mouse and you go from the head to the tail, you can recognize the lung, and then you go into the liver and you recognize the stomach here, and you see how this tumor here grows in this. And you can basically, conduct treatment studies where you can follow these tumors over time. And then last but not least, multiplex imaging studies, where we now perform immunohistochemistry with, in this case, 37 antibodies. And you can see the architecture of the liver that we recognize with the periportal areas. And depending on the question we have, we choose different antibodies. And we have done this in the past, where we used all these different markers to identify different immune cells, to identify different parts of the liver, endothelial cells, hepatocytes, etc. We can also then add functional markers that tell us something about the function of these cells, and then use this to study in-depth tumors and surrounding tissue. And this is just an example where you see this. We can basically zoom in into different cells depending on the marker that we look for. We can find all the hematopoietic cells. Here we see the liver sinusoids. Then we can find macrophages. We know there's many, many macrophages in the liver. We can find T cells. We can look at granulocytes, B cells, etc. So these are the tools that are at hand to really study in-depth tumors in the liver. And in the next few minutes, I'm just going to summarize and show you some of the results. So spatial immunophenotypes can actually predict clinical outcome in cholangiocarcinoma. This is a study coming out of Italy, where they looked at 192 patients and performed imaging of intratumor, invasive margin, and peritumor. And then performed also omics analysis, and they basically find different subgroups of patients. This is another study coming out of Shanghai, where they looked at the heterogeneity of the immune infiltration in intrahepatic cholangiocarcinoma, and then they compared it to established signatures. You see here the one from Daniela, for instance. I'm sorry. And these are really, really in-depth studies where you can really understand very well the tumor microenvironment. And then if we look at these, and this is just something because it comes, it's very interesting if we talk about immunotherapy, and it's going to come up later in my talk again. I just want to show you these immune structures called tertiary lymphoid structures, which actually appear to have some predictive value in immunotherapy response to cholangiocarcinoma. And you see these structures within the tumor, which basically consists of T cells and B cells. And then last but not least, of course, many different single cell studies where you get these beautiful, maybe sometimes confusing and difficult to understand cluster analysis and UMAP analysis here, where you can identify the different cells. And then once you have this, you can dive deeper in this. And here just showing you that the investigators have done this. And again, they have compared what do you find in the peritumor region and in the tumor region, and looked at different effector cells and different memory cells. And interestingly, in this study, and this is something similar to what we have found also in HCC, and I will come back to the cell type, and that's why I chose this, is the role of regulatory T cells, which appear to be very high within the tumor. And you know that regulatory T cells, they actually regulate, suppress immune responses, and are a type of T cell that we do not necessarily want to see within the tumor. And if we talk about gene signatures, I think it's also interesting to understand how do we take from a non-bioinformatic approach, how do we get there? So for instance, on the left you see a study where the investigators, this was Shara actually, who used this to look at rapid progressors and long survivors, and then she compared them to develop different signatures, and then obviously cholangiocarcinoma signatures based on immune responses or signs of immune infiltration or actually active immune responses. And similar studies have been done by us at the NIH in collaboration with my colleague and friend Xinwei Wang, where we also studied both liver tumor, HCC, and cholangiocarcinomas. So with this introduction about the tools that we have, and I use these tools all the time, I come to this conclusion: they are very descriptive. And I think that's always the problem I have. Yes, there is a subtype that's immune infiltrated, and there's T cells, but the problem for me is always where do we go next? If you want, remember the slide, if we want to improve the patient's outcome, we have to go into functional studies. And I love functional studies. I think if you don't understand mechanism and if you don't develop new therapies, you're doing something wrong here. So you have to do functional studies, and I'm going to show you some of those. What do we have in terms of really functional studies or immunotherapy studies in murine cholangiocarcinoma models? This is a study, or summarizing results from a study from Dan Duda from Harvard, who looked at the effect of chemotherapy, GEM-CIS, and how it actually changes the vasculature and then becomes very active in terms of inducing anti-tumor immunity in combination with CTLA-4 and PD-1. There is a study from Sumera that shows the immunotherapy with checkpoint inhibitors is only effective if you deplete myeloid cells and macrophages in this system, that the checkpoint inhibitors by themselves can not do this job. And in our own group, we have shown that we can get a potential synergistic effect when we use a MEK inhibitor, in this case, trametinib. If we think about cholangiocarcinoma specific, now this is the part that I mentioned when I talk about the specific mutations we have. I want to mention a study that also comes from a colleague of mine from the NIH, who's thought about the FGFR fusion, which you all know because we can target this specifically with FGFR fusion inhibitors. But what he actually thought is that these FGFR fusion are obviously also building specific epitopes that can be recognized by T cells. And then if this is the case, and you heard a talk about adoptive cell therapy earlier this morning, if this is the case and you can identify the T cell receptor that recognizes this FGFR fusion, you can develop this in a potential treatment. I think this is a very elegant approach where you specifically develop a treatment for this subset of cholangiocarcinoma. And then I know this was mentioned, so I'm not going to spend too much time on this. The very nice work from the Bardisi lab, where they looked at the effect of IDH efficacy or the effect of IDH inhibitors and how you can combine this, and their work actually has led to a clinical trial. You can see this that is currently ongoing, where ivosidenib is combined with nivolumab and ipilimumab in the second-line treatment of patients with IDH mutant biliary tract cancer. So this is just a slide summarizing where we are at right now. So there's adoptive cell therapy. There's obviously a lot going on with checkpoint inhibitors, potential vaccines. Bispecific antibodies are gaining more and more interest. I will tell you a little bit about agonistic checkpoint inhibitors that we're working, and then you already heard a little bit about these suppressive myeloid cells and how one can potentially target them. So now let's go back to immunotherapy trials in cholangiocarcinoma. So what do we really know what's happening in these patients? Is this immunotherapy really working? Well, I have to go back to this slide. This is the best clinical data. And then when I prepared my talk, and I realized there's not that much clinical result for this, and if you put in Google and search for emptiness, that's what you get. There's not that much known. We don't have a lot of correlative studies, and I know this is difficult, and I know in part why this is not being done, but unfortunately, in contrast to many other diseases, in cholangiocarcinoma, we know very, very little from our large randomized studies. And I know this is not because the investigators are not interested, because the investigators that run these studies are actually very interested, and they perform correlative studies in smaller cohorts. So Katie Kelly and Bridget Keenan, they did this study where they looked at the monocytes that express immunosuppressive chemokines in patients with biliary tract cancer that are associated with PD-1 resistance, and found that they can actually identify these patients, and that this could potentially display a resistance mechanism. In our own group, when we first started using tremelimumab, we looked at the very simple flow cytometry analysis, looking at CD8 cells and activation of CD8 cells. And yes, every now and then we find responders, but I have to be always very, very especially skeptical with my own data. So, this is helpful, but obviously sample numbers are too small, and I don't think this research or these studies go deep enough to really explain, why patients will respond or won't. And another very interesting study from Greg Lisinski's group and many in the group, and Nilo Azad, I think, was involved here, where again they used data and samples from a phase 2 study, combining a MEK inhibitor with PD-L1 and looked at different immune cells, and you always get a glimpse, that those patients that have activated T cells may be better, but unfortunately it doesn't really, it only helps us so much, and a lot of times, these are confirmatory studies. And last but not least, the study, again from our own group, from Dr. Monhill, who looked at our cohort of patients with pembrolizumab and oxaliplatin, again, now using tissue-based assay to identify T cells where, we find significant changes, or where we find changes. But if you're asking me what we should do, that is summarized here, I think we have to go way deeper. So we really have to do a very rigorous analysis that includes a very rigorous analysis of the medical data, the tumor biopsy. We can obviously use the tumor biopsy as a bulk or dissociate it, and then we can perform all these elegant assays, sequencing, spatial analysis. We can collect blood, whole blood, PBMC serum, can isolate these cells, can perform flow cytometry, sequencing, study exosomes, etc. You name it, and I'm going to show you some examples for this. You can look at imaging. So far, there's very little on imaging and diagnostic imaging and radiomics or immuno-PET. And then there's other body fluids, urine, stool, potentially ascites in patients with cholangiocarcinoma or bile, and stool samples if you are interested in the microbiome. Unfortunately, once you have all this data, many of us don't know, including myself, don't know anymore what to do with this. So you actually need to integrate this and using artificial intelligence and have very good collaborators that can actually help you do analysis, because it clearly goes beyond what you can do if you use the regular analysis that we have done and used in the past. All right. So with this short introduction, let me tell you how I see on what we are doing when I try to compare or translate from the bench to bedside and then go back. So The very first and very simple example is shown here. So here we looked at checkpoint inhibitors. You know we use PD-L1, we use CTLA-4, and then we decided, now let's use an agonistic antibody, CD40. And I think I've shown this here before, so I'm just going to keep it very brief. We used 3 different cholangio models where we have the tumor injected either subcutaneously, orthotopically into the mice, or we use one of these hydrodynamic models where we can use injection of oncogenes. And basically, you can see that this treatment is more effective than either of the treatment alone in all of these models. And we study the underlying mechanism, how this works. And Dr. Monne is now has actually opened the study. We opened the study last week and hope to enroll the next patient actually next week, or the first patient next week on this study, where we really directly translate based on findings that we obtained in mice and just on the side, the clinical part, as well as the mouse part, were actually supported by the Cholangio Foundation. So thank you to them, where we really translate, something and evaluate this. Now, we were one of the first groups to start looking at checkpoint inhibitors. And let me be honest, the only reason we did this is because we saw responses in HCC. And then I thought if something works in HCC, we had these early studies in HCC, and some of you may know this. We said, okay, so if it works in HCC, maybe, why not testing biliary cancer? So you see the first study, 2014, using tremelimumab in combination with local regional therapies in some of these patients, and the second study in 2016, where we used tremelimumab and durvalumab. I've already shown you the correlative studies for this were done. And to be honest, it didn't work. You find responses, you find immunological outcome, etc. But it was not something where, especially if you are in a clinic and you treat HCC patients and cholangio patients together, and you see there is a difference. You don't need high numbers of patients. You see, that there are differences. This is, what we observe when we perform, when we look at the combination of durva and tremelimumab. You can see here the cohort without ablation and with ablation. And the bottom line is, as I mentioned, every now and then you find some individuals that respond, and these responses may last very long, and we can look at responses and immune correlates. But the truth is, it's not what we're really looking for. So then, we had this brilliant idea of adding another drug to 2, which was anti-VEGF, because anti-VEGF, Avastin is being used for HCC, and that's how we developed these protocols. 1 doesn't work, 2 doesn't work, so then you tried 3. I mean, that's basically, this was my idea. And you see here the outline. So now we have basically, these studies, and we started this, and then something happened that we didn't expect. So we treated the first 5 or 6 patients, and I'm showing you just the result from these early patients, and you can see immediately we notice that there's something different. Now, you should not do this, and you're not allowed to do cross-trial presentation. You know what I'm showing you here? But I can tell you, you see that patients developed a response, and you see, they lived for a long time. And I'm going to show you, actually I'm not going to show you any more clinical data other than just one example of one patient. But this obviously caught our attention. It also caused our attention because this is the clinical course of one of the patients, and there's a lot of, I guess there's a lot of non-medical professionals in there. So I want to tell you what you can actually cause and do if you use immunotherapy. You can actually harm patients pretty significantly. So you see this patient actually developed a myocarditis. He developed a myasthenia gravis. He developed pharyngeal weakness. So in other words, he couldn't swallow anymore. He had double vision. He couldn't speak anymore. And he had issues with the heart. Okay, so, to make it easy. And I'm going to show you actually a picture of the patient with this consent. And, we had to really, deliver significant immunosuppression to this patient, to get him out of this crisis. And this was induced by immunotherapy. Immunotherapy works. If it works, it can really work. So this is the patient, and we ask him to look up, down, and then to the right or to the left, and basically the small muscles that you have to look to the right or left and move your eyes, they don't work anymore. You can see this. He basically, you don't hear this, that somebody's asking him, look to the right or to the left, and he can't do this. And we treated him then afterwards, and you can see he has his tubing because he couldn't swallow himself anymore. And now you see, how after the treatment, if you look at his left eye, all of a sudden it goes again, from the left to the right. And I'm just showing you this because I think it's a good example to see that, what immunotherapy is actually capable of doing. And obviously, it's also capable of inducing strong anti-tumor responses, and that's what we see in these patients. So I thought this was interesting. And I said, so let's understand why this is. What happened here? So then I went back to the bench. So you saw the patient, and now I said, let's look into mice. So we treated mice, similar to what I have shown you before, either with the combinations of PD-L1 and CTLA-4, or VEGF and CTLA-4, or nothing, and you can see that the combination of the three drugs, PD-L1, anti-CTLA-4, and VEGF, is the most effective one. And then we said, okay, so this is basically, and the other ones, other than the combination of anti-CTLA-4 and anti-VEGF that is not really widely used in patients, wasn't as effective. But now we have the advantage that we actually have a model where we can study this in more detail. So the first thing we did is we looked at the tumor-infiltrating immune cells, and we performed this complex analysis, and basically what you can see here is the following. So most of you, I'm sure, are familiar with these t-SNE plots where you see different types of cells. So this is shown here, and there is one specific plot up here, this pink one, and then that is interesting because this one actually only appears in the condition where we use these three treatments. You don't see it in any of the other 5 conditions. There's another population. Look at this green population. You see this green population here is here, and it is here, it is here, it's maybe not as strong here, and here it's gone. Okay, and obviously there's other ones that change. So these 3 populations, they really change, and there's a third one that is a little bit more difficult to show. But you can really, with these type of assays, start, to dive deeper, and that's what we do. So we looked now in the same model again, and now we limit this only to double and triple treatment. So double is anti-CTLA-4 and anti-PD-L1, and triple is the same with anti-VEGF. And you see that the treatment is more effective, tumors are smaller, but we also find more CD8 cells per gram tissue only with the triplet. But we also find Tregs. Now, if you remember, I told you very early on, Tregs are not so good because they actually suppress immune responses. So this caught our attention. It says, how can this be? It's so effective, but at the same time, we have actually a cell type that is very immunosuppressive. And I will come back to this in a second. Now, because we saw B cells, this pink plot, we got very interested in B cells because most people always talk about T cells, checkpoint inhibitors, CD8 T cell, it's straightforward. So we started looking more at the effect, on B cells. And what we did is the following. So one experiment is we took out B cells out of the game. So we used an antibody for CD20, so we take out all the B cells. Same result. Down here, triplet is effective. We take the B cells away. It doesn't work anymore. Now, where do the B cells come from all of a sudden? There are certain cytokines and factors, and there's a growth factor. It's called BAF. Indeed, we find BAF in the tumor secreted in the triple therapy, but we don't find it in the double or in the control groups. And if we take away BAF in the system, the treatment is not effective anymore, suggesting that we start the treatment, we get BAF, and then we get the B cells, and this starts to work. Now, where is the BAF coming from? This is shown here. We basically, now do an assay. It's called a prime flow, where you use actually, where you can actually use a tool to look for an mRNA by flow cytometry, because some of these cytokines are not that easy to detect by antibodies. So we now use a tool to actually identify the message within the cell, but it's a flow-based assay. And you can see this number of BAF-positive CD45 cells is higher in the CD45 group, both at a relative and absolute number. And then we look at CD11b positive cells, CD3 cells, and the double negative cells. So double negative means they're neither CD3 or CD11b. And you can see, where the BAF is actually coming in this particular setting. If we look at the tumors in these mice, we find tertiary lymphoid structures. So these are the tumors, and I had mentioned this earlier to you. You see all of a sudden there are, if you look at an H&E section, these immune conglomerates, and if we stain them for CD3 or for B cells, B220, you see they stain very highly, suggesting that there's such a strong immune response that you actually induce these TLS. Now, what about the Tregs? Remember, the Tregs, they don't fit. It just doesn't make sense that you get more Tregs. So we started to look at the Tregs in more detail. And the way we do this is that we look at their function and we look at specific transcription factor. And without going too much into the details, what happens is that we actually change the phenotype of the T regulatory cells into what we call a fragile Treg, which is actually starting to release a lot of interferon gamma, becomes more interferon gamma, which is becoming an effector cell and loses its suppressor function. We can also test this if we isolate these cells from these mice after treatment. We see that they are actually, if we use an assay in vitro, where we look at the suppressor function, that these cells all of a sudden are no longer as effective in suppressing T cell responses as they are in the beginning. Now the question is, how is this being induced? And there is data in the literature that you can change this phenotype of the Tregs by a cytokine, IL-12. And we went back to our mouse model, and now in this case we now blocked IL-12. The same thing, you already know this, treatment without treatment, the triple treatment, and now we take away the IL-12, and you can see the efficacy, the gain that you have by adding the anti-VEGF is gone. Both here, if we look here, or at the tumor weight. Now the next question is, where is the IL-12 coming from? Again, we look at the IL-12 positive cells using the same type of assay, and it suggests here that the majority of the IL-12 is actually coming from B cells. So you remember I told you, we actually first get the BAF, we get the B cells, now we have the IL-12 that leads to the Tregs, and that ultimately leads to the strong immune response. Okay, so this was difficult for the non-immunologists, I'm sorry. But the interesting thing is that there's another notch to add, because VEGF can actually also work directly on Tregs. So what we did is we used a conditional knockout where we knock out the VEGF receptor in FOXP3 positive Tregs, and if we use mice and inject tumors, the tumors don't grow that well anymore, indicating that indeed these regulatory T cells lose their suppressor function. Now, if we combine these mice, and add double therapy, you can see that this is actually very effective. Again, suggesting that this effect of the anti-VEGF is actually twofold. It's on the Treg directly, and then also through this B cell mediated mechanism. Okay, now we go back to the patient, because what I didn't tell you yet is that obviously we collected a lot of specimen from these patients. We collected blood specimen, we collected tumor specimen from these patients. So now we can go back and see, is what I've shown you in the mice and what we believe is happening and explaining this extraordinary and very strong immune response, actually also can we see this in our patients? And again, I'm only going to show you some of the most important data. So you've seen this before, and because we have the samples from our patients from the prior trial, we can actually again do the same comparison. You remember, double versus a triple therapy. So these are the changes in immune cells. Let's start on the left. So this is data from flow cytometry, where we look at the different immune cells in these patients, and you can see which ones go up, and Tregs go up, monocytes change, and B cells change. There's not a lot of change in the B cells. If we look at all the B cells, we find some in the tumor infiltrating, some changes here. So at least this significant change in the Treg and change in the monocytes appears to be very similar to what we have seen in our mice. Now, if we look at chemokines, we have the change of chemokines pre versus post, and post is basically 4 weeks on treatment. On the top, you see the difference between, for the patients receiving the double therapy, and down here, the ones receiving the triple therapy. The first thing we see is that VEGF goes down, which is something that you expect because we use an anti-VEGF antibody. But what we also see is a clear increase in IL-12, which we don't see with the double therapy, a clear increase in BAF that we don't see with the double therapy. And remember, these cytokines were required to see an effect in our mouse models, along with, a couple of other chemokines, some of them related to B cells. So obviously you will never get a one-to-one, but I think there's a strong indication that similar things that we have seen in our initially in the patients and in the mice, you can be confirmed here. And last but not least, and we also find actually these TLS structures in our patients. So all of a sudden we find immune infiltrates in these tumors from these biopsies from these patients. And then last but not least, we performed single-cell sequencing on these PBMC, and we looked at the regulatory T cells in more detail. And if you zoom into the regulatory T cells and you find the different clusters of natural Tregs, I mentioned fragile Tregs or atypical or unstable Tregs, you can see, and then look at pre versus post, you see that all of a sudden these Tregs exactly change the same way we would predict from our mice data, that the natural Tregs post-treatment actually go down, and the unstable or fragile Tregs go up. So this, I think, I hope, I was able to convince you, this is an example where you really go, you make an observation in patients, and, you collect the samples from these patients, and then, you try to use this to understand, and then obviously with the ultimate goal, to improve, the treatment even further and develop this more. So let me summarize. So these are changes that we have seen with this triplet therapy, and I've mentioned some of them. I didn't mention all the changes, and the mechanism is basically shown here, that we believe, that upon treatment, you get T cells to release interferon gamma, and BAF, which then changes B cells to release IL-12, change these Treg type, and then these also make interferon gamma, ultimately increasing the anti-tumor immunity. And obviously there are changes by the anti-VEGF also on the monocyte compartment. Okay, so now in the last 10 minutes, I'm going to talk about the microbiome, and I have given microbiome talks before, so I'm not going to repeat this. I'm just going to give you a brief highlight about this, what we're doing here. So a number of years ago, we looked for the first time in liver tumors, and we saw that antibiotic treatment is actually reducing growth of liver tumors. And we identified the mechanism, which was published. And again, I'm not going to repeat this, but we saw that this is mediated by bile acids and then changing, again, immune cells, in this case, NKT cells in the liver. And based on these findings, we developed a clinical trial and said, if this is true, can we find something else? Can we find this in patients? And that is what is shown. So we performed a phase 2 study using nivolumab and vancomycin in patients with liver tumor disease. And unfortunately, and that's why I have Donna Mabry, who is a nurse practitioner working here, this fell straight into COVID. You can see this is why usually we don't run around like this, which obviously, if you do these correlative studies, made it a little bit more difficult. You know, Donna is here, so if you want to recognize her, this is how she really looks without the mask. But we were able to enroll 22 patients in this particular study. We didn't see significant clinical responses, but we collected samples from these patients, peripheral blood and stool samples and tumor samples. And what you can see here, this is just a high-level view of the gut microbiome. And I think it's very clear to see that before vanco and after vancomycin, you know, this is completely different. We also looked at the bile acids and have, you know, more data to come from this study. We had another study that was now directly related to cholangiocarcinoma, where we saw that the gut microbiome can change myeloid-derived suppressor cells and how they are being recruited to the liver using different antibiotic cocktails and in the context of cholangitis or liver disease. But last but not least, and this is the coolest, it's the microbiome-brain connection. Okay, so what do I mean by microbiome-brain and tumor connection? So for those of you, you know this immunity cycle, you know this that has come out, that where we know, or the cancer, sorry, the Weinberg's cell cycle, you know, with the different factors that control tumor growth. And if you have read the latest issue of Cell, this has actually become a little bit more complex. And there are actually, interestingly now, also mentioned a neural cancer crosstalk and the macro and tumor microenvironment. Now, if we go back, or if we check our cell phone, you can see the vagus nerve is actually the body's secret superhighway. And if you go back to 1859 or 49, the introduction to the study of experimental medicine, it was, I didn't touch it. It's such was the experiment case, who repeated my experiment of the floor of the fourth ventricle to cause artificial diabetes. Okay, so they did an experiment where they changed and manipulated the brain, and they induced the diabetes in this model, suggesting already many years ago that there is this brain-liver interaction. Now, there is different mechanisms that have been described how nerves and tumors can potentially interact directly on the tumor cell, but there is also this, I think, very, very interesting field of neuroimmuno-oncology. Now, even more interesting maybe is a study that was published in 21, where they looked at 49,000 individuals that had a perforated peptic ulcer, and they either underwent regular simple suture or a truncal vagotomy. And then they followed these patients and did matching, and they found that actually those patients that underwent a truncal vagotomy were less likely to develop liver tumors. Okay, Dr. Gors is starting to laugh. He doesn't believe me anymore and says this is a wrong study, but I will convince you how it works. So we did the same in mice. Okay, so we perform either a sham vagotomy or a hepatic vagotomy. So we cut the branch of the vagus nerve that goes into the liver and inject a tumor cell into these mice. And you can see that if we look at the tumor growth by bioluminescence or at the tumor weight, there's a clear difference that a hepatic vagotomy clearly impacts the growth of liver tumors. If we look at the different immune cells in the liver, and these are just the different players, you can see that they vary. There's a difference in the CD8 T cells. There's a difference in the CD8 T cell function only in the liver. We don't see any changes if we look at the spleen. And if we perform this experiment and deplete CD8 T cells, you see that there's no longer an effect, suggesting that indeed this experiment where we perform the vagotomy really relies on the presence of CD8 cells. Okay, I only have a few minutes left, but that's why I can only show you a few points of this. In these vagotomized mice, we also performed actually behavioral studies And we could see that actually once you have a vagotomy, they are actually less fatigued. And that brought us to the following experiment, and this is the last experiment that I'm going to show you here. So here we use mice, and either we do a sham injection into their liver, or we inject a tumor. And we wait for 3 weeks. After 3 weeks, these mice are euthanized, and we collect the stool from the cecum of these mice. Now, we take a new set of mice. We make sure that they get used to the environment. We perform a test where we look how active they are, a behavior test. And then we perform an FMT, so we give them PEG. That's the same solution that you use for colonoscopy to clean the gut. And then we either transfer only the stool sample. There's no tumor in this experiment. The stool sample from a tumor-bearing mouse or a tumor-free mouse. We repeat this several times, and then we perform these behavioral studies again. And you can see, if we look at the sleep and inactivity, that there is a clear difference, that the mice that actually received the tumor samples are clearly more active. The stool samples from the tumor-bearing mice are clearly active. What this suggests is basically that the tumor signals or changes the gut microbiome, and then that the gut microbiome signals up to the brain and affects the behavior. So again, I only showed you very little of this because I don't have more time, but I still was excited enough to, I wanted to show this. So we show that hepatic vagotomy can reduce liver tumors. This is CD8 mediated. I didn't show you this, but they also had reduced fatigue. I've shown you that the gut microbiome can, from tumor-bearing donors, can sufficiently alter the behavior. And then we performed experiments where we actually show that this also leads downstream to changes of immune responses in the liver. And I think this is interesting because it also brings up a completely new approach, because now you can potentially use acetylcholine drugs that work on acetylcholine receptors to potentially treat patients. So with this, my time is over. I don't want to stop before thanking the Cholangio Foundation and everybody. It's so beautiful. This is the reason why my talks was not ready. It was this morning. This picture, this is a wonderful event. It's always fun to come here. It's fun to talk here. I need to thank my group, clinical group, lab group doing these studies. I get obviously a lot of support within the NIH from extramural collaborators, from some funding institutions that support on top of the intramural funding I have, including the SITC. And with this, I'm happy to take 2 questions. It's 56. Thank you.
Questions for Dr. Gretton? Dr. Gores, do you want to redeem yourself?
So you can see I was paying close attention.
I didn't doubt that.
Yeah. In the liver transplant scenario, of course, the liver is
yes,
innervated.
I know.
And you get recurrent tumors, and they're quite, I understand there's a confounding effect of immunosuppression and other things, but it's clearly not a dominant mechanism in that setting.
There is no question that this is not a dominant mechanism. I mean, I just want to make sure that it's not. And this is basically just the beginning, to start new avenues, how you can do this. And again, I didn't show you this, but we actually use conditional knockout mice where we specifically knock out the acetylcholine receptor 3 on CD8 T cells, okay? And if you grow a tumor in these mice, they grow a little less. And I said a little. It's not a lot, it's significant, but they grow less. Now this shows you, if you are able to manipulate an acetylcholine receptor on a T cell, there is certainly more that you can do. And that's basically the concept that I'm trying to describe. And I'm not saying that this is a major mechanism, but regardless of microbiome, regardless of vagus nerve, etc., we can actually bring it down to one receptor, which is an acetylcholine receptor on a CD8 T cell, and that leads to a biological difference. Okay.
Well, you have one more question.
Thanks for the great talk. And a question about the anti-VEGF and enhanced checkpoint inhibitors. And you showed a mechanism through IL-12 and the BAFF. So I'm just kind of curious, kind of one level upstream, kind of what's causing that, right? Is it because the anti-VEGF potentially repair the leaky vessels, or enhance the, immune cell penetration or TLS formation that triggers that, or it's some effect on endothelial cells that probably, yeah, so would love to hear.
So remember, what we're doing is we're actually using always a doublet. So you already have T cell activation. So I think, and the mechanism that I describe is in the presence of checkpoint inhibitors. It's not the VEGF, the anti-VEGF alone doesn't do this. So you need to have already an activated immune response. You already need to have CTLA-4 in the game to change, to start this mechanism. And I don't think it has anything to do with permeability. It's just basically, the cytokine milieu and interferon gamma that you find by activated T cells that jumpstart this.
And seeing something in the lymph nodes, or?
Possible, yeah.
Great, and I still cannot open it, but you can hear me. Thank you very much. We're ready to move on to the next session. It's now time for session C, Recent Advances in Cholangiocarcinoma Biology. It is my pleasure to introduce the moderators of this session, Dr. Gregory Gorse and Funda Meric-Bernstam. Dr. Gregory Gorse definitely needs no introduction. His research contribution include over 3 decades of federal funding, 750 publications, and he has contributed to establish many protocols regarding liver transplantation for cholangiocarcinoma. Definitely, as I said in its no introduction, the second moderator is Funda Meric-Bernstam, who is the chair of the Department of Investigational Cancer Therapeutics and also the first speaker of this session. She is a clinical trialist with a focus on decision support for precision oncology, biomarker-driven clinical trials, and antibody-drug conjugates. Thank you both for moderating this session.
Thank you. It's really my honor to be here today. And thankfully, I didn't have enough slides, so I can just try to figure out if we can turn on the slides first. Oh, yes. Here are my disclosures. As mentioned, I run the phase 1 program, so we have a lot of research funding that supports clinical trials. Some funding for preclinical modeling as well. And today I'm really just going to talk a little bit about FGFR2, inhibition in cholangio, and what we know about mechanisms of acquired resistance, so we can kind of strategize. And this is relatively informal, and we'll discuss at the end in a panel discussion how together we can work to understand this target better and how we can target it better. As you know, FGFR2 is really probably the one of the most important successes we've had at a cholangio community, and I really just want to acknowledge the importance of the Cholangiocarcinoma Foundation to make this a successful enterprise. We recognized that this is a important driver in cholangiocarcinoma, and I have to say that I had really no awareness of even cholangiocarcinoma until FGFR fusions became a thing, and I was invited to give a keynote talk on genomics at the Cholangiocarcinoma Foundation, and I really just fell in love with the foundation and the community, the sort of collaboration between the scientists and the clinicians. But really that awareness of the importance of genomic testing to identify targets that were still investigational in cholangiocarcinoma led to this international success in the multiple drug approvals in cholangiocarcinoma overall, but especially in FGFR2. This, as you know, of course, is a target especially important in intrahepatic cholangiocarcinoma. And, it is variable based on study frequency, and obviously initial studies maybe overestimated how important or how frequent it is. But really that excitement that we saw with the first series when infigratinib data came out, where we saw these responses that were quite durable in a disease where we had nothing, leading to the initial sort of successes with targeted therapies in cholangiocarcinoma. This, of course, was followed by the data that emerged with pemigatinib. This is a reversible inhibitor of FGFR2, and as you know, that data that was published in Lancet Oncology showing that in 35.5% of the patients that have either FGFR2 fusion or rearrangement, having these deep responses with almost all of the patients getting these agents, having disease stabilization or responses, really showing how exciting this target is. And then more recently, the approval of futibatinib, which is an irreversible FGFR inhibitor, and this data published in New England Journal around the time where the FDA approval of this agent, again, coming in for FGFR2 fusions and rearrangements. So we're all very excited to see this data. This is definitely a genuine target. We are aware, I think, as a precision oncology community that fusions are special. There are very few mutations or amplifications that have the durability of targeting a fusion. But to be fair, FGFR fusion, inhibition doesn't have the same durability that we've seen, for example, with TRK inhibitor responses or lung inhibition with ALK fusions or ROS fusions. And although most of our patients have had benefit, ultimately resistance develops. And that is really what we need to talk about as a community and try to figure out, okay, this is successful, but how do we do better? So earlier today, we were talking about liquid biopsies, and I just want to talk a few minutes about this because this is a tool that we're all excited about, clearly used differently by different treating physicians and investigators. I just want to, highlight that, again, we need to work together to try to better get the data we need to be able to actually systematically use liquid biopsies in our care. As you know, cholangiocarcinoma has a decent amount of DNA shedding. We do believe that most of the time we can detect mutations and fusions in the plasma. The fusion detection by liquid biopsies are very variable based on the assay, and that's really important to also to know. Now, liquid biopsies are especially important in cholangio because we don't always have as much tissue to be able to do assessment, but they're not a substitute for tissue biopsies because there are several other genomic alterations that need tissue for identification, like deletions. And we have emerging targets such as MTAP that need us to start looking at those kind of targets as well. In addition, there are several cell surface targets that are emerging in cholangiocarcinoma, so we still need tissue. So my emphasis on this slide isn't because liquid biopsies are all that, a substitute, but importance of it because A, we can use it maybe to assess early responses, but importantly, we can use this to understand mechanisms of acquired resistance. In investigational trials, we often do longitudinal sample collections, starting on treatment, early time points like after cycle 1 or after cycle 2, and subsequently after progression. And those after progression studies that I'm going to show in a few minutes are really giving us importance of acquired resistance. But as a community, I think we need to figure out how we can pool resources to look at the early dynamics to show that that actually has clinical value, so we can eventually make that actually a time point that is billable and will benefit our patients in their care. So, at the time where the reversible FGFR inhibitors were in clinical trials, really interesting data from Lipica and the very strong translational group from MGH highlighted that there were emerging mutations in patients that got FGFR inhibitors. And these were a variety of different mutations in the same patients emerging. And when you see liquid biopsy results and you see multiple mutations in the same gene or in the same pathway, you know that that is like a real phenomena of convergent resistance. So, they were able to publish this really elegant paper in Cancer Discovery showing patients where they had, ctDNA detection of tumor genomics, and then upon response, decline the variant allele frequency, and then increase in not only other mutations, but the recognition that new FGFR2 mutations were emerging as a resistance mechanism, along with nice data showing that those mutations conferred resistance to the FGFR inhibitors. This is important to understand, but of course better if you have something to do when you see those data. So around that time, futibatinib had gone into clinical trials, and a few years later, they were able to update their studies now, showing that there were patients where there was a response on an early first-generation agent, resistance, and they were able to challenge with another FGFR inhibitor and show again a response. And that was accompanied with really elegant studies with longitudinal sample collections demonstrating that there was a drop in variant allele response at the time of response, emergence of resistance, and then upon re-challenge, there was a response of those mutations that had conferred acquired resistance to the first inhibitor, but not to the second inhibitor. These kind of studies are still evolving and not really, we haven't maybe done a very good job in incorporating it into our care, but it's really important to start looking at that data coming from these papers because they highlight that if different inhibitors have different efficacy on different mutations. And this is relevant because now we're seeing patients that have a variety of different genomic testing platforms in their hands. They've had liquid biopsy with this, a variety of different mutations. So we really need to be able to start incorporating what do I give next, knowing the sensitivity and resistance pattern to these mutations. So, along with obviously Western blots, et cetera, also demonstrating that, in some agents you have inhibition of FGFR signaling while within the context of some mutations, but not in others. All of these preclinical studies, you see tables like this. I'm sure many of you are familiar. Essentially, green means you're sensitive, yellow means you're so sensitive, and red means you're resistant to that specific, in the context of that specific mutation. And this was data that was incorporated into the New England Journal paper for futibatinib, and you can see at least of the mutations detected here, futibatinib was quite effective across a variety of different acquired resistance mutations as well, although the New England Journal paper was based on a series of patients that were FGFR inhibitor naive. So, there are now several other drugs in development. The kinase data that was developed, for example, which inhibits FGFR 1, 2, 3, demonstrating that it had greater efficacy in the context of some mutations. Although for business reasons, this agent is not going forward. Another agent, in development in this space is the agent from Tyra. And then there are also selective inhibitors of FGFR2. The thought is that maybe we can push the dose harder if we don't have the side effects and as much hyperphosphatemia seen that's associated with FGFR1 inhibition. And again, you can see in green, some of the mutations that are still resistant to futibatinib now with the relay agent also having sensitivity and early signals of efficacy with these agents. A really nice paper reviewing all of the published data to date was in Clinical Cancer Research this year, where they looked at the spectrum of acquired resistance mutations detected on ctDNA upon progression in 82 patients from 12 different published reports. In these series, 49 of the 82 patients, or 60%, had a detectable secondary FGFR2 mutation, one or more of them. And the most common mutations were the N550 molecular break or the V565 gatekeeper mutations, representing 63% and 47% respectively. And these kind of studies are very helpful, as I said, because I think that systematically looking at which drug is active in the context of each mutation is important. And in our own effort, we have a precision oncology decision support program, and we're hoping to be able to start incorporating this information so that when we see patients, we can better select our next step. There's still a lot we don't understand. There are other agents like multikinase inhibitors that also hit FGFR that show efficacy in the progression setting, whether that is all FGFR related or whether it's because of the angiogenic immune modulating other effects of these agents, that's not clear. Especially these agents, like the FGFR inhibitors, none of these studies have had systematic on-treatment biopsies to really give us insight into what differentiate those patients that respond and don't respond. But early on, 2017 reviews already signaled that like with other receptor tyrosine kinase inhibitors, they could be other mechanisms of resistance. You can have upregulation of other RTKs. You can have other mechanisms to activate PI3 kinase or MAP kinase signaling. For example, even in Lipica's first paper that I published, they had a really elegant autopsy study where they saw multiple different sites had multiple different mutations. And not just FGFR2 mutations, they saw PTEN loss and other alterations in PI3 kinase signaling, suggesting that downstream signaling activation may confer resistance. In our effort, we did this retrospectively, looking at our patients that had gotten either tissue or liquid testing. And actually, if you go to the paper we published in Journal of Hepatology, the figure before this is very complex and embarrassing in some ways because it shows how disorganized we are about our tissue and liquid testing, because people ordered whenever they thought it would be very beneficial. So different platforms were used, and it makes it a lot more complex to understand what's really happening. But bottom line is, if you start looking, we saw multiple patients starting to develop not just FGFR2 mutations, but MAP kinase mutations. For example, if you squint and look at panel C, you can see, there are patients that have multiple mutations in the MAP kinase pathway, multiple different KRAS mutations, an NRAS mutation, a BRAF mutation, really highlighting that, we know that FGFR signaling predominantly signals through MAP kinase signaling, and those cancer cells need that. So in a little bit more organized fashion in the phase 1 trial with futibatinib, when we realized the trial didn't have liquid biopsies, we collected at Anderson serial samples, and we saw that there were patients that on futibatinib as well had generated FGFR mutation acquisition upon progression, but also we demonstrated that multiple hits on the MAP kinase. So we had one patient, for example, who had seen an FGFR inhibitor before, came in with FGFR2 mutation, still had a response. That highlights that we may not be able to, we can't just presume a patient has a FGFR2 mutation, they're not going to be responsive. But there was one patient who had multiple different MAP kinase mutations and had a baseline NRAS and BRAF mutation that increased upon progression and then acquired several other mutations, BRAF, NRAS, 12C as well as 12D. Preclinically, when we took MAP kinase mutations and we put them in cholangiocytes, indeed we were able to show that when we put a fusion, we sensitized the futibatinib. When we add a KRAS mutation, we make this resistant. And it's not just simple as, we can just add like a MAP kinase inhibitor now and make it sensitive. Though there's preclinical in vitro synergy, this was not sufficient to cause regression in this setting in vivo. So we need to do a better job from the get-go. And then I talked mainly about acquired resistance, but we need to understand adaptive responses better as well. When we treat tumors, they have an early response to an upregulation of survival signaling. And as I mentioned, almost all of these studies have been conducted without on-treatment biopsy, so we don't really understand what is happening to the immune environment or as well as cell signaling to see if we can better select combination partners for those in these studies. But elegant study that we'll talk about, I think, later today as well, highlights that EGFR signaling is at least one mechanism of survival signaling that gets upregulated. And this concept at MGH is being brought into a clinical trial now. So I just want to say this is a time where we understand more what we don't understand. We don't understand it enough to treat patients that much better. I think we really need to start talking better about co-alterations and when we can use that information for treatment selection. Clearly, different FGFR inhibitors have differential activity in different mutations. We need to figure out how we can select patients better based on that. Liquid biopsies, I think, are very justifiable upon progression. We need to figure out whether there's a role of early sampling as well. And there's definitely need to better treat patients get more effectively from the get-go. So we need new mechanism of action and new combinations. This kind of work takes a team. I'd like to thank everybody in my department, Investigation Cancer Therapeutics, GI Medical Oncology, as well as my lab. And thank you for inviting me.
So we'll move on to the next speaker, which is Dr. Liberalis Boila, who will be speaking on identification of novel vulnerabilities in mutant IDH-driven intrahepatic cholangiocarcinoma. Dr. Boilis is at the Fred Hutchinson Center, works with Dr. Kugel, and who's carrying on the work of Dr. Saha. Both Dr. Kugel and Saha trained with Nabil Bardisi, went to the Fred Hutchinson Center, and they're continuing on that legacy. Welcome.
Thank you so much to the organizers for the opportunity to present our work. Today I'm going to tell you a story around our recent efforts to identify novel vulnerabilities in mutant IDH-driven intrahepatic cholangiocarcinoma, hereafter referred to as ICC. The Kugel Lab is a basic and translational oncology lab with a focus on trying to understand the biology of pancreas and biliary cancer. Our mission is to discover and mechanistically define novel therapeutic strategies that can be validated in preclinical cancer models with the ultimate aim to translate these lab findings into new clinical trials. As many of you may know, the biliary cancer lab at Fred Hutch was started by Dr. Shub Saha. After his untimely death, Sita took over the lab and his work and our vision. So most of the work being presented today is a continuation of his efforts stemming from his initial hypothesis and his vision to cure ICC. So you have been hearing since the morning about these genetic mutations that are found in cholangiocarcinoma. So cholangiocarcinoma can be divided into distinct genetic subtypes based on the underlying mutation signature. Mutations in isocitrate dehydrogenase, or IDH1 and 2, are found in approximately 20% of all ICC patients. IDH is a metabolic enzyme. The wild-type IDH converts isocitrate to alpha-ketoglutarate in the Krebs cycle, whereas mutant IDH converts alpha-KG into 2-HG. 2-HG is an oncometabolite. It inhibits the function of alpha-KG-dependent dioxygenases, many of which are epigenetic enzymes, like the KDM histone demethylases and the TET family of hydroxymethylases. So this causes widespread epigenetic changes, which often drive tumor formation. During his postdoc, Shub found that the IDH mutant ICC was specifically sensitive to Src family kinase inhibitors. Shown on the left is the sensitivity assay showing two IDH1 mutant ICC lines to dasatinib and siracatinib, and their sensitivity is similar to that of the highly sensitive CML cells. And dasatinib is already clinically approved in CML for BCR-ABL positive patients. He validated this using the cytotoxic assays showing that the IDH1 mutant ICC cells, shown in red, was much more sensitive than the IDH1 wild-type cells, shown in black. And we'll be following this color code throughout the rest of the presentation. However, surprisingly, this sensitivity was only restricted to ICC, as the IDH mutant chondrosarcoma or even lung adenocarcinoma did not show this sensitivity. So we have been trying to understand that what is the specific mechanism and why is only the IDH mutant ICC hypersensitive to dasatinib. To do this, we have developed relevant disease models. We have our panel of human 2D cell lines, and additionally, in collaboration with clinicians at University of Washington, we get patient samples from which we make organoid cultures, tumor slice cultures, and PDXs. We have also developed several novel genetically engineered modified mouse models and orthotopic tumor xenograft models, orthotopic and subcutaneous tumor syngeneic graft models. I'm happy to announce that this work has recently been accepted for publication in Science Translational Medicine, and this work was led by a very talented postdoc in the lab, Iris, and a technician, Caroline. To start off, we first tried to look at SRC-associated signaling pathways after dasatinib treatment. So shown here are 3 IDH wild type and 3 IDH mutant lines, and we saw a consistent and specific downregulation of phospho-S6K and phospho-S6 upon dasatinib treatment. Now, S6K phosphorylation is known to be mediated by mTOR. However, we did not see any changes in upstream or downstream signaling components of the mTOR pathway. We also did not observe any other changes in other downstream targets of SRC. To assess the impact of S6K activity in our IDH mutant cells, we first looked at cell size by flow cytometry. So this was done after 24 hours of treatment with dasatinib before any apoptosis can set in. And we saw that dasatinib treatment caused a reduction in cell size, specifically in the IDH mutant cells. As a second readout, we also looked at global protein translation via the puromycin uptake assay. So puromycin, it's an analog of the 3' end of the amino-acylated tRNAs, and it gets incorporated into newly synthesized peptides. And we saw that dasatinib treatment caused a reduction in puromycin uptake and global protein translation, again, specifically in the IDH mutant cells and not the wild-type cells. To confirm that dasatinib is indeed acting through inhibition of SRC, we used SRC gatekeeper mutant lines. Now, these gatekeeper mutants have a mutation at SRC T341 residue, which makes the SRC binding site inaccessible to dasatinib. So dasatinib cannot bind to or inhibit SRC in these gatekeeper cells. And we saw that these gatekeeper cells had much lower apoptosis upon dasatinib treatment, and they also showed a rescue of the phospho-S6K and the phospho-S6 levels after dasatinib treatment. As further validation, we used another pair of isogenic gatekeeper lines, and herein also we saw that there was a rescue of S6K and S6 phosphorylation without a change in any of the other signaling components. The gatekeeper lines also showed a rescue in global protein translation after dasatinib treatment. As further validation, we knocked down SRC using independent shRNAs, and we saw that SRC knockdown mimics dasatinib treatment. It causes a reduction of phospho-S6K and phospho-S6, again, only in the IDH mutant lines. So to summarize, we see that dasatinib treatment inactivates SRC. This causes reduction of S6K and S6 phosphorylation, leading to reduced protein translation, cell size, proliferation, and survival. However, SRC is a tyrosine kinase, and S6K and S6 phosphorylation, they take place at serine and threonine residues. So we hypothesized that there must be an intermediary molecule involved in the signaling pathway. To identify novel targets of SRC, we conducted a phosphoproteomic screen. We wanted to see specifically those phosphopeptides which were downregulated in the IDH mutant cells, but not in the gatekeeper cells after dasatinib treatment. And so this led us to the identification of the top hit being a protein called MAGI phosphorylated at tyrosine 373. MAGI is known as membrane-associated guanylate kinase family with inverted domain structure 1. It is an intracellular adapter protein and has several WW and PDZ protein binding domains, along with a catalytically inactivated guanylate kinase domain. Now, MAGI has been known to have inactivating mutations and hypermethylation in different cancers. So classically, it has been described as a tumor suppressor. However, ICC patients usually do not have mutations in MAGI1. There are 3 isoforms of MAGI1 based on their splicing sites, and these differ mainly at the C-terminal regions and their subcellular localization and their function. And MAGI1C, which has a nuclear localization signal, is the predominantly expressed form in ICC. We next wanted to see the function that MAGI is playing in IDH mutant ICC. So we knocked out MAGI1 using CRISPR, and we see that it leads to reduced apoptosis after dasatinib treatment. Similarly, knocking down MAGI1 using an siRNA, again rescued the S6K and the S6 phosphorylation after dasatinib treatment. So this indicates that MAGI1 is necessary for the S6K and S6 phosphorylation reduction, and it acts as a negative regulator of this pathway. To confirm that MAGI1 Y373 is indeed a SRC substrate, we used several mutants of MAGI1. So the Y373 resides within the second WW domain of MAGI1. And we overexpressed SRC and either the wild-type MAGI or a MAGI Y373F mutant that cannot be phosphorylated. And we see that only when the wild-type MAGI is present, we get phosphorylation at Y373, and we don't get it with the mutant MAGI. Next, we overexpressed SRC with either the full-length MAGI or truncation mutants of MAGI. And again, we see that loss of the WW domain is leading to a decrease in phospho-MAGI Y373. Next, we did immunoprecipitation experiments with SRC and either the full-length MAGI or the truncation mutants of MAGI. And again, we see that the loss of the WW domain is leading to decreased binding of SRC to MAGI. So we found that SRC binds to MAGI1 at Y373, and this prevents the reduction in S6K and S6 phosphorylation. However, MAGI1 is an adapter protein and has no known kinase activity towards serine and threonine residues. To our knowledge, no other kinase apart from mTOR has been known to phosphorylate S6K at the T389 residue, which we see here. And we observed no change in mTOR signaling after dasatinib treatment. So we hypothesized that a phosphatase, namely PP2A, might be involved in regulation of S6K in our system, as it is a known mediator of the T389 mark. So PP2A is a phosphatase. It is a heterotrimeric protein consisting of a scaffold, B regulatory, and C catalytic subunit. To confirm if PP2A is indeed catalyzing the dephosphorylation of S6K, we used ocodeic acid, which is a potent PP2A inhibitor. So we treated our IDH mutant cells with ocodeic acid for different time points, followed by dasatinib treatment. And we see a rescue of both S6K and S6 phosphorylation with deoxyribonucleic acid treatment. So this shows that dasatinib is indeed working through activating the PP2A phosphatase activity. To further validate, we used siRNA against the PP2A catalytic subunit. And here again, we see that knocking down the PP2A catalytic subunit rescued phospho-S6K and phospho-S6 after dasatinib treatment. PP2A activity is regulated by two post-translational modifications: phosphorylation at Y307, which is usually considered to be an inactivating mark, and methylation at L309, which is usually considered to be activating. And phosphorylation of PP2A at Y307 has previously been shown to be mediated by SRC. We saw that SRC inhibition using dasatinib led to a reduction of phospho-PP2A. Again, this happened only in the IDH mutant cells and not in the wild type or the gatekeeper variants. So this suggests that SRC inhibition is indeed promoting PP2A activation, which is then dephosphorylating S6K. To summarize, we found that SRC phosphorylates MAGI at Y373 and PP2A at Y307. This prevents PP2A from dephosphorylating S6K. Upon dasatinib treatment, SRC is inactivated. There is loss of phospho-MAGI and phospho-PP2A. PP2A now gets inactivated and can access and dephosphorylate S6K. So MAGI being an adapter protein, we wanted to next see if it is indeed bringing together a new signaling complex. And using a number of immunoprecipitation experiments, the details of which I'm not going into now, we saw that MAGI brings together SRC, PP2A, and S6K into one signaling complex, facilitating S6K dephosphorylation. Therefore, we have defined a new signaling complex in IDH mutant ICC. Upon dasatinib treatment, the PP2A phosphorylation is lost. It gets activated, has access to S6K, dephosphorylates it, leading to reduced phospho-S6, reduced protein synthesis, cell growth, and survival. So from all of this data, it is evident that S6K and S6 signaling are the downstream effectors, or rather the final targets of dasatinib. And interestingly, we observed that there is reduced phospho-S6 at baseline between in the IDH mutant cells compared to the IDH wild-type cells. We further validated this in primary patient samples, wherein the IDH mutant patients show a lower phospho-S6 at baseline. Indeed, inhibiting mutant IDH using AG120, which is a small molecule inhibitor, it increased phospho-S6 levels both at baseline and maintained it after dasatinib treatment. So this indicates that a functional mutant IDH is needed for dasatinib to work. We next generated dasatinib-resistant clones and found that even these clones had elevated phospho-S6 at baseline. So this led us to hypothesize that phospho-S6, that low phospho-S6 is acting as a marker of dasatinib sensitivity, and reducing phospho-S6 may be improving dasatinib efficacy. And to achieve this, we combined dasatinib with this molecule, DIACC3010, which is previously known as M2698. So this is a dual AKT S6K inhibitor, which has recently been shown in the phase 1 clinical trial to be effective for advanced solid cancers. So using this combination in our in vitro studies yielded really promising results. We saw that the combination was more effective in reducing phospho-S6 compared to dasatinib alone. Next, we tested this combination in vivo using IDH1 wild type and IDH1 mutant PDX models. And interestingly, we saw that the combination significantly reduced tumor growth in the IDH1 mutant PDX. Now, this IDH1 mutant PDX was derived from a metastasis from a patient who was unresponsive to AG120. And therefore, we are really excited seeing this data that this combination can become a third line or perhaps even a second line treatment option clinically for ICC. The combination treated mice also showed enhanced survival after drug withdrawal. So based on this study, we are now planning a phase 1 and 2 combined clinical trial, which will be led by Bill Harris and Gentry King at Fred Hutch. The phase 1 will consist of a dose escalation study to find the maximum tolerated dose of this combination, which will be used as a recommended dose for phase 2. Looking forward, the studies here has led to several exciting future directions which we are now interested to explore. So MAGI1 is a scaffold protein, and we hypothesize that MAGI1 has more binding partners than only PP2A. So using a proximity labeling technique which employs TurboID, which is an engineered biotin ligase, we plan to identify novel and transient binding partners of MAGI1. And for this, we have already generated a TurboID MAGI1 fusion construct and confirmed its expression in IDH mutant ICC. After biotin labeling, the proteins will be eluted using a streptavidin column and subjected to LC-MS to identify the binding partners. Next, since we saw that PP2A activation is taking place after SRC inhibition, we are now interested to see how the PP2A is differentially regulated between IDH mutant and IDH wild-type ICC. So as mentioned, the PP2A L309 methylation is the activating mark, and it is regulated by two enzymes, LCMT1, which is the methyltransferase, and PME1, which is the demethylase. So we are going to study these and other enzymes in the regulation of PP2A in the IDH mutant ICC. And finally, we want to identify novel combination therapies with AG120. So AG120, which is the mutant IDH inhibitor, has recently been approved as a second-line therapy. And this was based on the phase 3 clinical trial, which showed overall improvement in overall survival. However, the partial response was only 2%. And so this underscores the need to increase the efficacy of AG120. Unfortunately, our results indicate that dasatinib cannot be used with AG120 since AG120 treatment makes the cells resistant to dasatinib. So as an alternative approach, we have conducted a screen using a library of 133 FDA-approved drugs, and we have identified several top hits from here that we are now validating in additional models. I would like to take this opportunity to thank my mentor Sita for her unwavering support, and to Shub, without whom this would not have been possible. I thank all my lab members, especially Iris, who has been driving the IDH projects. I would like to thank all our collaborators, the Fred Hutch core services, our funding agencies, and finally the patients and family members who give meaning to the work we do. If you have questions or are interested to know more about our research, I would be more than happy to answer it at the end of the session. Thank you.
Just keep you up here because we're going to have a panel discussion at the end. And I'd like to invite Dr. Dill up. He's our next speaker. Michael Dill is a physician scientist and hepatologist. He's interested in primary sclerosing cholangitis and cholangiocarcinoma. He got his MD PhD from Basel, University of Basel, Switzerland, and did a postdoc at Carnegie Laboratory in the Harvard Medical School. I'm told it's better if it's on. He'll be, he'll be talking about some of his work. I'm really excited to hear about.
Yeah, thank you very much for the introduction. As also thank you very much the organizers and the Cholangiocarcinoma Foundation for inviting me here. It's my first time here, but I have to say, so far the session has intrigued me a lot, and I think it's very, very nice to have this condensed kind of exchange and of this common interest. So my title of the talk is Innovative FGFR2 Fusion-Driven Mouse Models, and just to start, I'm a physician scientist. These are my disclosures. I don't have to introduce that to you, but just to quickly clarify, we're gonna talk about FGFR2 fusions, which we primarily find or exclusively find in intrahepatic cholangiocarcinoma, and depending on which cohort you're looking at, this is about 10 to 20% of the patients. And this is also data that you have been presented before, about 30 minutes. I want to keep it short, but just the two main clinical trials that had led to approval of these drugs, pemigatinib and futibatinib, as second-line treatment for FGFR2 fusion cancers, with overall objective response rates of about 35 to 43%, while there's also, I think, very interesting that we have additional compounds that have been tested or are under development right now in clinical trials. And this is a similar graph you have seen before. So we have now, with these next generation inhibitors, also the chance to retreat patients that have acquired resistance mechanisms. And I try to summarize this here a bit. This kind of, leads us to this clinical challenges that we find. So far, we haven't had any kind of combination trials with FGFR2 inhibitors. Is there anything that speaks for treatment synergies? One of the unsolved questions is, should we treat sequentially, so start with a first-generation inhibitor and then go on once they acquire resistances, or is it better to immediately go to a broader inhibitor? All these kind of things we have to address eventually. This has also been the subject of several talks already, predictors of response, biomarkers. So far, we have no evidence that the fusion partner of FGFR2 plays any role in the function or the response to the treatment, but there's a lot of co-alterations. This is a follow-up study from the FIGHT trial, where they looked at the co-alterations within this patient cohort that was treated with pemigatinib. And as you can see here, there is no clear patterns of co-alteration, genetic changes that might predict response or non-response to these or better or slower response to these tumors. And we also, when we look at this, obviously we get, we're talking of a subtype of a subtype of a subtype. So at some point, we're getting to very, very rare cases where it might be just very difficult to address in a clinical fashion. Just an additional finding from that trial, the only thing, when we combine not single genes, but basically tumor suppressor genes as a whole, that has been shown, not surprisingly, that this had led to a bit, worse survival outcome. So my point is here that we, one approach probably should be looking at preclinical models, and I tried here in this slide to kind of summarize some common models that are being used, and especially indicate the limitations these models have. Like every model, there are pros and cons for which one to use. The classic ones, the germline edited ones, obviously are not so practical anymore because they're just still time consuming to produce. It's also sometimes difficult to control tumor burden. What is very commonly used, therefore, is in vivo gene editing. People have shown it to you, my previous speakers. One very common way is hydrodynamic tailwind injection of transposon-based systems. The thing is that if you want to create cholangiocarcinoma out of this, it usually requires oncogene transcripts that are driving hepatocytes into a cholangiocyte lineage to create cholangiocarcinoma. So that is one of the drawbacks of this system. And then the most genetically complex systems are obviously using direct patient material, but again, here we are limited by immunocompromised hosts that we need, so immunocompromised mice typically. Another system that has been arising over the last years is due to the fact that we now can culture cholangiocytes as organoids. So in an unlimited fashion, there's gives of time to edit them, genetically engineer as syngenic lines, but again, this is of course an implantation model. So this is kind of a short overview of the spectrum of systems we have. And now I quickly want to present you FGFR2 fusion models that have already been published. There are two main ones that I've picked out here. One from the group of Zaborowski in Hanover. They used a transposon-based system where they delivered 3 different oncogenes or tumor suppressors into the liver. This is so it was basically p53 knockout, KRAS, G12D mutation, and an FGFR2 fusion. They delivered that by electroporation into the liver, so into hepatocytes. And one of the, at that time, it wasn't better known when they started the project, but KRAS G12D, as you might know, is not very common to find as a co-alteration with FGFR2. So that is one of the drawbacks of this model. And they used a human transcript. You heard from Daniela Sia that this is, was the limitations of these first models, that they used a human transcript. And while this was, elegant to look at mechanistic models in terms of resistance in a cell intrinsic matter, we cannot really analyze the tumor microenvironment very well. And this is another model using wild-type organoids and genetically engineered them, introducing also a human fusion transcript and also p53 knockout. And again, they had to use immunocompromised mice to create an in vivo model. And then, of course, you have just seen this morning, for those of you who have attended, that Daniela Sia developed a method now, also a transposon-based model with a mouse transcript, which I think is really the way to go if we want to look at the microenvironment. We have chosen a bit of a different approach, where we also try to generate a syngenic model, but using an endogenous fusion. And I will show you, let me quickly go here first. The system we're using, that is kind of our tool, our working horse. So we're using cholangiocyte organoids from a wild-type mouse. This we can culture indefinitely, and then we can sequentially introduce genetic alterations. We initially started as a proof of principle with some known and strong oncogenic mutations, and I'll show you this in this slide here. So these are just some examples of these models. So basically, the first one you can see here is p53 KRAS SMAD4. Let me say like this: p53 KRAS by itself is sufficient upon implantation to create cholangiocarcinoma. They all express strongly cholangiocyte markers. I didn't show you here now other markers, stromal markers, but I think you can appreciate there's a lot of stromal reaction in these tumors that have glandular patterns very reminiscent to human cholangiocarcinoma. And just as an example, so we can, are very flexible with this model. So here, another one with ARID1A deletion, which is also commonly found. And then we also created a MSCI high tumor by deleting MSH2, so one of the mismatch repair genes. So we wanted to do something similar for FGFR2, so let me quickly jump back here. Just what we knew at the time when we started this project was from the Silverman study, which were the most, most abundant data available. So we selected some of these genes that are commonly co-altered with FGFR2 and tried to create these models. So just a very, very brief reminder, FGFR2 receptor is working basically by dimerization upon binding of the ligand FGF, which then leads to phosphorylation and continuous or activation of the path of different downstream pathways. So if you have a fusion partner, this leads to a continuous dimerization and therefore continuous activation of the pathway. So to create an endogenous system, we used CRISPR-Cas9, targeting the FGFR2 where it typically fuses with other proteins, and then we used one of the most common ones here. This is the gene AHCYL1. But again, as you heard, there are more than 60 different ones. And then we created also a single DNA template to basically facilitate the fusion in the system. So this is the work line we chose at that time because it's commonly been used in all these models, p53 knockout as well, because it just facilitates carcinogenesis, and it also makes these wild-type organoids grow a bit faster, so it's usually easier to work with them. And then we transfected them with CRISPR-Cas9 plasmids. So in the end, we will then have an endogenous fusion, so there is no additional transcript, there is nothing additionally added to the genome. And then we functionally select these cells by withdrawing them of growth factors. This is just to show you that we then could confirm in these clones that we indeed have a fusion transcript, both on the genomic DNA level and also on the messenger RNA level. We could also see on the protein that it somehow starts to become overexpressed, probably might be more stable. This is then, we created a library of different of these, we call them PFFs, so FGFR2 fusion. And we created a library of these 4 common tumor suppressors that are associated with it. And in general, most of them grow similarly well in vitro. The only one that is a bit slower in growth or reduces in growth is BAP1, which we don't really know why, but has been described before also by other groups. And the point here I want to make is, so when we have these cells in vitro and we withdraw them from growth factors and we compare the fusion organoid with our gold standard, let's say the KRAS mutated organoid, we don't really see a difference in growth or anything. However, when we implant them, similar to what has been shown this morning, PFF alone was not sufficient to induce tumors. Could be plenty of reasons. We're looking into it. So one, but we believe must be some cell extrinsic reasons. Maybe it's also P27. I got inspired this morning thinking about that. However, as I told you, we had created already all these other lines, and when we systematically tested them, the one that was most sufficient to induce tumors in these mice, and again, these are untreated mice, these are just wild-type mice, so we are not in an inflammatory environment. That's something we're going to test if that might facilitate tumor growth. Nevertheless, in this we had an 80% tumor penetrance upon implantation, and you can see here again typical features of cholangiocarcinoma, glandular patterns, and also we see a lot of immune cell infiltrations here. The other line that led to tumors as well was P16 knockout, albeit with a lower penetrance. And importantly, in this system, P10, so we wanted to rule out that P10 is basically driving this here, but P10 alone in this implantation system was not sufficient under the same conditions to form tumors. So we don't think that's the point, and FGFR2 fusion here really is driving to some extent. So to conclude, to my knowledge, this is the first time that we have generated an endogenous, so within the genome of the mouse, this fusion, and which we can use as a preclinical model to further study aspects of this disease. Interestingly, although we do not see a big difference in vitro upon implantation, there is differences compared to KRAS G12D in this system. We're checking this by using immunocompromised mice if it's an immunogenicity issue or something else. And we're currently working on analyzing these immune populations, and obviously would want to use that for doing synergistic drug testing in this system. With that, I would like to conclude. I need to point out Nugo here, who did a very talented medical student who did a lot of this work, and of course all the other people in my lab, my funders, and my collaborators. Thank you very much.
last speaker for the session is Dr. Pedro Miguel Rodrigues, who's at the BHRI in San Sebastian. Dr. Rodrigues trained with Dr. Cynthia Rodrigues and Professor Rui Castro in Lisbon, and then went and worked with Dr. Benalis, and is now directing his own group on metabolism and cancers and cell cancer, and has a large interest in biomarkers and for diagnosis in cholangiocarcinoma. So welcome.
Good afternoon, everyone. Thank you very much for the kind introduction. And first of all, I'd like to thank the organizing committee, Daniela and Sumera, and also the CCF for inviting me to be here today in such an inspiring meeting. So now it's time for biomarkers. Some people already mentioned the importance of biomarkers. So in this talk, I'll talk a little bit about the emerging biomarkers for the early diagnosis and also for the risk stratification of patients with cholangiocarcinoma. Oh, I did something that I shouldn't. Okay. So as you all know, the diagnosis of cholangiocarcinoma may be an incidental finding in up to 25% of cases, after usually the appearance of unspecific symptoms such as jaundice, fever, weight loss, fatigue, or abdominal pain. And how is the diagnosis of cholangiocarcinoma achieved? It's usually achieved by combining distinct approaches such as imaging techniques, with the measurement of serum non-specific tumor biomarkers such as CA19-9 or CEA, with tumor biopsy, which is usually mandatory to confirm the diagnosis. However, we know that imaging techniques and non-specific tumor biomarkers are somehow suboptimal diagnostic biomarkers because, firstly, they difficult the differential diagnosis of malignant and benignant tumors, and they do not usually allow to differentiate the two types of primary liver cancer, or even the identification of mixed tumors. On the other hand, tumor biopsy is accurate, although it's invasive, and there are several inherent risks, such as bleeding, dissemination, infection, among others. So therefore, there is an urgent need for accurate non-invasive diagnostic tests for cholangiocarcinoma. So why no CA19-9 and CEA are no useful diagnostic biomarkers? So in this first study from the International Cholangiocarcinoma Registry, we have shown that CA19-9 and CEA are barely elevated in patients with early cholangiocarcinoma stages and are more increased in patients with advanced disease. And actually, we observed that increases in the levels of both these biomarkers are more highly associated with patients with locally advanced disease or metastatic disease rather than patients with early disease. And in fact, we observed that having an higher an elevation of CA19-9 or CEA or both elevations are highly associated with patients to present metastatic disease. Moreover, we know that CA19-9 is not present in up to 10% of the population due to the presence of inactivating mutations in the FUT3 gene, which leads to the absence of CA19-9, and therefore it raises some concerns on the use of CA19-9 as a biomarker for cholangiocarcinoma. Rather than being a diagnostic biomarker, CA19-9 was shown to have a prognostic value, along with the ECOG performance status and the disease status. It was shown in the NSCA registry that it has an independent prognostic value in cholangiocarcinoma. And actually, other studies have shown that decreases in CA19-9 levels during radiological treatment are associated with a better outcome in these patients. And therefore, CA19-9 might be better used as a prognostic and surrogate biomarker rather than a diagnostic biomarker. So PSC, primary sclerosing cholangitis, is now the best well-known risk factor for cholangiocarcinoma. And we know that the cumulative incidence of PSC cholangiocarcinoma is more or less 15%. These patients are usually diagnosed in the 40s or 50s when compared to cholangiocarcinoma of other etiologies that usually appear more later on after in the elderly. More importantly, we know that cholangiocarcinoma in these patients usually develop in the first year after PSC diagnosis. And it's very difficult to diagnose because there are a lot of overlapping symptoms and features between PSC and cholangiocarcinoma. So how is the surveillance of patients with PSC done? Usually an MRI and MRCP is conducted every 6 or 12 months in these patients, and once a dominant stricture, dominant mass, or increased CA99 levels are detected, an ERCP is suggested along with breast cytology to try to detect some polysomy that might indicate tumor development. Nevertheless, these patients have a very poor prognosis with a median overall survival of less than 10 months in unresectable cases, and it's actually the leading cause of PSC-associated mortality. So the major question in this population, patients with PSC, is that can we predict if the patients will actually develop cholangiocarcinoma? And for example, when doing imaging procedures, if we see a biliary stricture, can we detect if this stricture is benign or malignant? So basically, we need accurate non-invasive biomarkers that will allow us to firstly predict which patient will develop cholangiocarcinoma in order to develop surveillance and early detection programs. To do this, liquid biopsies have been very useful in the last years to try to find novel biomarkers. This is a minimally invasive approach and includes the isolation and analysis of cell-derived material, such as, for example, DNA, RNA, extracellular vesicles, and circulating tumor cells that can be detected in blood or in other body fluids. And these liquid biopsy have different applications in cancer, since they may have the screening of cancer, the early diagnosis. It may also allow patient stratification, guide therapeutic decisions, monitoring the response to therapy and the appearance of minimal residual disease, and also predict the relapse and the prognosis of these patients. And different approaches have been done in the last decades regarding liquid biopsies. In this talk, I will focus on 2, which is the ones that we have been studying in a lab. So firstly, I will address extracellular vesicles. So what are extracellular vesicles? They are small membrane spheres that are secreted by all the cells into the extracellular milieu, and they are present in all the biological fluids, such as blood and urine. And they are categorized according to their origin, their size, their molecular composition, and their biological function. And inside these vesicles, and also in the membrane, there are different proteins, metabolites, nucleic acids, and lipids that might be very useful to discover new biomarkers. So in a study that our lab conducted almost a decade ago, we isolated serum extracellular vesicles from patients with HCC and cholangiocarcinoma, and we evaluated the proteome of these serum extracellular vesicles to identify novel biomarkers to first early and specifically diagnose both HCC and cholangiocarcinoma, and secondly to try to distinguish both HCC and intrahepatic cholangiocarcinoma. And as you can see here, we were able to identify several protein biomarkers that were useful for the diagnosis of HCC, for the diagnosis of cholangiocarcinoma, and also for the differential diagnosis of intrahepatic cholangiocarcinoma at HCC. We now moved and wonder if these serum extracellular vesicles could be also useful to detect biomarkers to identify cholangiocarcinoma in patients at higher risk, such as patients with PSC. So our aim was to identify biomarkers for the early diagnosis of cholangiocarcinoma in patients with PSC, and also try to identify predictive biomarkers that could allow us to predict and stratify patients with PSC according to their risk for cholangiocarcinoma development. In this case, we use patients with all patients were confirmed by histology or cytology, and for PSC patients, we included only patients that we know that have not developed cholangiocarcinoma in more than 5 years during follow-up. So we follow the same strategy that we have developed before. So we obtain serum from these patients. In this case, in the discovery cohort, only healthy individuals, patients with PSC, and patients with PSC-associated cholangiocarcinoma. We isolated the extracellular vesicles, and we measured the proteomic profile of these vesicles. As you can see here, we obtained the typical round shape of the extracellular vesicles that were enriched in markers of EV, such as CD81 and CD63, that were absent in all cell extracts, confirming the purity of our EV fraction isolation. When we evaluated the proteomic profile of these vesicles, we identified distinct biomarkers that allowed the diagnosis of PSC cholangiocarcinoma when compared to PSC and healthy individuals that had high area under curve values that were even improved when we selected only patients with early stages. So among these all biomarkers, we then asked ourselves, are these biomarkers only helpful to diagnose cholangiocarcinoma in patients with PSC, or can they be used to all cholangiocarcinomas independent on disease etiology that we call the BUN cholangiocarcinomas? To do so, we recruited more samples from patients with PSC, from patients with PSC cholangiocarcinoma, and now we included cholangiocarcinoma from different etiologies that are now PSC, and included more samples with from healthy individuals. We isolated again the serum extracellular vesicles and measured the proteomic profile of these vesicles in order to try to identify the biomarkers. While we observed that among all the biomarkers that we identified, some of them were specific for the diagnosis of cholangiocarcinoma on PSC, others could only diagnose cholangiocarcinoma in patients without PSC. Almost all of the biomarkers that we identified were considered BUN cholangiocarcinoma biomarkers that could allow the diagnosis of cholangiocarcinoma independent on disease etiology. So among them, these BUN CCA biomarkers, we now wanted to validate this with a technique that could be used in clinical routine. So among these pan-CCA biomarkers, we selected specific biomarkers to be validated according to the increased abundance. So we selected the biomarkers that were increased in patients with cholangiocarcinoma, and we selected among the best diagnostic biomarkers and with the ones that were commercially available, reliable ELISA kits. So before doing the ELISA, we tried to identify if these biomarkers could be detected in total serum, therefore avoiding the extracellular vesicles isolation steps. So to do so, we did an immunoblotting of serum EV fractions of patients with cholangiocarcinoma and healthy individuals, and also with serum that was depleted with extracellular vesicles. And as you can see here, among these 7 biomarkers that we tested, all of the biomarkers were almost enriched in the EV fraction, but also detected in serum. And if we now include patients with the 3 subtypes of cholangiocarcinoma and healthy individuals, we were able to observe that the levels of these biomarkers were increased in total serum for patients with cholangiocarcinoma compared with healthy individuals, therefore validating our findings. So now we move to ELISA, and we validated the levels of these biomarkers in our cohort, and we were able to validate the proteomic results, in which we found that the levels of all these 7 biomarkers were increased in both PSC cholangiocarcinoma and cholangiocarcinoma when compared to healthy individuals and PSC patients. We then combined these biomarkers to try to improve their diagnostic capacity. So we divided our cohort in a training cohort comprised of 70% of the patients, and we generated a model that was then validated in the testing cohort and also in a cohort including only patients with local disease. And for example, we observed that the combination of only 2 biomarkers, CRP and FRIL, allowed the diagnosis of cholangiocarcinoma independent of disease etiology with a Harry under the curve, or 0.92, that was even improved when considered only patients with local disease, obtaining an AUC of 0.94. These findings are now under an international validation in a cohort of patients from Latin America, from the Escalon project. And some preliminary results have shown that even in patients from Latin America, we are able to observe that these biomarkers have some diagnostic capacity. And we are currently recruiting samples from not only patients with PSC and cholangiocarcinoma, but we are now including samples from other risk factors of cholangiocarcinoma, such as gallstones and cirrhosis, and also including patients with HEC to do the differential diagnosis of cholangio and HEC. And if you have any samples or want to collaborate in the study, please contact either me or Jesus. We will be happy to provide further information. Next, we asked ourselves if we could also predict CCA development in patients with PSC before the patients developed the tumor or before the patient was clinically identifiable. And to do so, we used another cohort of patients with PSC that we call PSC2-CCA, that are patients that we know that developed cholangiocarcinoma during follow-up, but the sample was obtained before any clinical evidence of malignancy. And as you can see here, among the 7 biomarkers, we observed that 4 of them were already increased before any clinical evidence of malignancy. And when we observed the alterations of these biomarkers, we observed that patients with PSC that presented alterations in the levels of these 4 biomarkers, 100% of these patients developed cholangiocarcinoma during the follow-up, while patients without any alteration on these biomarkers did not develop any cancer during follow-up. We then combined these biomarkers in panels to increase their capacity, and we observed that, for example, the combination of the 4 biomarkers provides an area under the curve value of 0.92 that was significantly superior to CA99, the current gold standard for CCA diagnosed in patients with PSC. Can also these biomarkers allow the differential diagnosis of intrahepatic cholangiocarcinoma and HCC, and we observed that all the 7 also allowed to differentiate these 2 types of primary liver cancer, with the combination of the 2 of them provided an area under the curve of 0.96. Apart from extracellular vesicles, we are also interested in studying metabolites, since tumors are highly metabolic rich tissues, and these alterations might be detected in serum. And we published a study some years ago in which we tried to use serum metabolomics to identify new panels for the diagnosis of HCC and intrahepatic cholangiocarcinoma. And we did this with a platform in the Basque Country in Spain, which is All Metabolomics, which is a company that is highly specialized in serum metabolomics and in the development of diagnostic tests for liver diseases. And what we observed is that there were 104 metabolites that changed in patients with HCC compared to control, with some metabolites providing very good area under the curves values. The same was true for intrahepatic cholangiocarcinoma compared with control patients. And when we compared patients with HCC and intrahepatic cholangiocarcinoma, we obtained the same results with some metabolites changing. And this allows us to develop different models. And for example, the combination of 6 metabolites allow us to differentiate HCC and intrahepatic cholangiocarcinoma in discovery and validation cohorts with area under the curve value higher than 0.9. Can we also now predict the development of cholangiocarcinoma in patients with PSC using serum metabolomics? And to do so, we are conducting a multi-center international study in which we included almost 500 patients with PSC from 11 healthcare centers from 8 different countries. This is a study that was conducted in collaboration with several members of the IPSC study group, PSC Partners, and PSC Support Funding. And we observed that in both discovery and validation cohorts, there are already different serum metabolomic alterations in patients with PSC cholangiocarcinoma that were maintained when we consider only patients with early PSC cholangiocarcinoma. And we, considering a diagnostic model with including only 10 metabolites, we were able to have a diagnostic model with almost 1 of accuracy for the diagnosis of cholangiocarcinoma. Finally, since it's not only liquid biopsy, we are also interested in trying to identify tissue biomarkers that might allow us to predict recurrence after tumor resection. And in this case, we did a literature revision to identify potential prognostic biomarkers that may need to be validated when identify 49D6 biomarkers that we evaluated their association with survival doing a Cox regression analysis. And now with a cohort of 210 patients from different institutions, we are now validating these results by an open array technology, trying to identify some tissue biomarkers that would allow us to predict recurrence after surgery. So overall, we saw that there are novel non-invasive strategies that are emerging in the search of biomarkers which may add value for cholangiocarcinoma. Increased CA99 is more associated with disease dissemination, with prognosis, and might be considered a surrogate marker. And we now need to validate the biomarkers that were identified in serum with ELISA. For different panels might be validated in the future. The same for serum metabolomics, also for the differential diagnosis of ICCA and HCC, and also for the diagnosis of cholangiocarcinoma in PSC. And we are conducting now international prospective validations to try to move these panels into clinics. With this, I would like to thank our research group in San Sebastian, led by Professor Jesus Banales, and all the institutions that fund our work, and thank you very much for your attention. Thanks.
Okay, we have half an hour for discussion, which is really awesome. I wanted to see if there's any questions from the audience. Yes, I see a hand. Go ahead. Microphone is coming. Greg wanted to run today.
Thank you. So I have two questions for Dr. Dill. So I may miss this, but I just wondering, have you tried whether those new developed endogenous FGFR fusion mouse models is sensitive to the FGFR inhibition? And the other one is how similar they can mimic the observation of immune signature in patients.
Yes, so this is all work in progress. So we're testing that because we don't know, right? Because these inhibitors, we think they're very conserved, that area, but we have not really tested that yet, to answer your first question. And your second one, that's true, we have not systematically analyzed all the immune cell infiltrates to make sure that, you know, we have obviously it's a very important thing to do, yeah.
But along those lines, as you get that data, how do you quickly transition that to the clinic? Do you have, you know, other than this venue, do you have venues to make sure we're aware of which confers resistance and what would be a combination strategy?
I mean, we have the facilities to test that. We do that routinely in the clinics. So I think, yeah, we could set that up to do that, yes.
We're very excited to see your results.
Got another question for Michael. Everybody's wanted to know about the models. So one of the
we can't hear you.
Sorry, everybody wants to know about your models. So one of the things that in the metastasis literature is that one of the adaptations that the cells need to reimplant. I'm just thinking about when you're adding additional drivers in. Doesn't implant, it'll grow, it grows nicely, but it doesn't implant. One of the adaptations is being able to handle reactive oxygen. And there's some data that some very kind of simple techniques of N-acetylcysteine, things like that, can buffer the cells enough to essentially tolerate the implantation, and we're certainly playing around with that a little bit. Do you see any gain of function or other cell adaptive gains when you add in the other drivers?
I mean, usually what we've done mostly there, you know, once we have one of these classic drivers, right, that are really driving an oncogenic signaling pathway, and okay, P10 is one story, but the others are usually the ones we've tried are common, you know, are not these classic, you know, that they're pro-proliferative pathways, like or targeting SWI/SNF complex, etc. So we haven't really looked there into detail, but for sure, everything, you know, I'm happy to exchange later to find some strategies, maybe improve. Also, I didn't mention that, but I mean, if you use 2D cell culture, you know, usual implantation of cells is around 1 million, or, you know, so here we're usually a bit lower, 200,000 to 500,000, because obviously it's very painful and expensive to expand these organoids. That's one of the drawbacks in this system.
Thank you. So mine's a clinical question for Pedro. Thank you for your presentation on the biomarkers, particularly in PSC. And I think, obviously, from a clinical perspective, the most important time to identify somebody with a cholangiocarcinoma who you've screened is somebody who's got early stage disease. Have you been able to show a stage dependency on your biomarkers?
So thanks a lot for the question. It's very interesting, and we try to, at least the ones that we validated by LASA, we stratified a cohort in early and late stages, and there were no differences. So these differences are really occur early in tumor development. We try to check a little bit the function of the proteins to try to understand what could be happening. Most of the patients are, the proteins are acute phase proteins like CRP, for example. But there are some, for example, OE3, it's named oncoprotein-induced transcript, so it's a transcript that is induced in early stages of malignancy. So we believe that these biomarkers are already altered in the beginning of the disease, and they do not further increase. So we did not detect any differences according to the stage.
Pedro, can I follow up on that?
Yeah.
Given that some of these are acute phase reactants, and with PSC and even with cholangiocarcinoma, you can get segmental obstruction, you can get subclinical cholangitis. Do you have any other, I mean, CRP would suggest that that's going on. Are you controlling for serum bilirubin levels, other markers of cholestasis, that type of thing?
So we correlated the levels of these biomarkers with bilirubin, with ALT and AST, and with alkaline phosphatase, and we didn't see any correlation on these biomarkers. So we think that they might not be associated with that. What we now want to do, and we actually applied for funding for that, is now to conduct a prospective phase, because all of these cohorts were retrospective. So now the idea is to collect, do a prospective validation in which we recruit patients with PSC. At recruitment, we try to evaluate the levels of these biomarkers, and at the beginning stratify them according to the risk of cholangiocarcinoma development, and actually find if these biomarkers could allow us or not to detect cancer.
Pedro, it looks like you're predominantly focused on diagnosis. Have you identified anything that's a potential novel target? And are you using any of these tools for therapy response monitoring or target identification of existing targets?
That's our next step. So one of the biomarkers, at least one of the ones that we validated by ELISA, which is PHAR, we know that it is only expressing cholangiocytes in the liver, so it's islet specific. We are now doing some evaluations in tissue. We know that is increased also in both in PSC cholangio and cholangio, and we validated the levels in cholangiocarcinoma cells in vitro compared to normal human cholangiocytes, and it's highly increased. And there is already a study with this protein in HEC, although it's not expressed in hepatocytes, it somehow gains the expression when tumor develops. And it's an oncogene, so we believe that this protein might have an important role in cholangiocarcinoma. So there are other, not the ones we validated by ELISA, but among the list, there were other proteins, because I didn't mention, but there was also some EV proteins that had a prognostic role, and some of that were associated with immune response and might be predictors of immunotherapy response. But we want to evaluate that in the future.
So I have a question for Liberalis, and great talk, everybody. I apologize. I grew up as a signaling person, and so I'm trying to get all of your signaling network into my head all at once. But I believe you didn't talk about, MAGI-1 has an mTOR site at serine 148. Is there the potential that that would lead to like a feed-forward or a feedback inhibitory mechanism that might also sort of magnify the effect that you guys are seeing in your therapeutics?
Just to understand your question, you're saying mTOR phosphorylates MAGI-1?
At serine 148.
Yes. During the screen, we looked at mostly the phosphotyrosine peptides, and the focus of that was because we are inhibiting SRC, and that's why we focused only on the phosphotyrosine peptides, and that's how we arrived at the Y373. But now we are expanding our study of MAGI, and we are trying to see other interacting partners, other post-translational modifications. And so I think this is there in the future, like we are trying to expand our study of MAGI-1, and definitely this is one aspect that we are planning to look into.
Also for Liberalis, so you described the mechanism of dasatinib and SRC inhibition, but do we have an understanding of why the IDH mutation confers that vulnerability? Is it something about the epigenetic state of the cell?
So that is, I think, the question that we want to understand. Till now, we have identified the mechanism, we have identified certain pathways. We don't see any differences in total MAGI-1 expression between the IDH wild type and IDH mutant. We have done in vitro SRC kinase assays. We don't see any difference in SRC activity between IDH mutant and IDH wild type. One hypothesis which we have is that PP2A may be intrinsically more active in the IDH mutant cells. And so to keep PP2A in check, we need a phosphomagi. We have not validated this hypothesis, and currently we are trying, that's why the next step is to identify PP2A activation between the IDH mutant and IDH wild type, how the PP2A activity is controlled. But right now, we don't have an answer for that, and that is something we are looking into. Yes.
Yeah.
Others have shown other Src kinases are activated in cholangiocarcinoma. And I was wondering, is it specific for Src itself, or we've seen LCK, for example, be activated? Have you looked at other Src kinases?
So this was actually answered in Shoup's original paper, where he did gatekeeper mutants of different Src family kinases, like FIN, and he showed that it's only the gatekeeper Src which is conferring resistance to dasatinib. So, and we also did knockdown assays, and we confirmed that it's only specifically Src which is acting and not other Src family kinases.
You started your IDH discussion by saying that the drug was approved with with 2% rejected response rate. The in vivo data you showed really had very modest growth inhibitory effect with dasatinib. I was curious whether you've seen actual regressions and how many models you've tested for IDH.
For the PDX data, we have here just the data of one wild type and one mutant, and we are trying to expand our IDH1 mutant PDX cohort in order to have more data points. But as you said, the dasatinib did show a modest effect, and the combination, the main goal of the PDX was to study if the combination is having a more profound effect than dasatinib alone, which we did indeed see. But definitely, we are expanding our number of PDXs, and I think that will be able to better answer this.
You were looking at a AKT S6 kinase dual inhibitor for your studies. There is a AKT inhibitor now approved for breast cancer. I was curious if you looked at that in combination as well.
We actually studied 3 different S6K inhibitors, out of which like the AKT S6K dual inhibition was it was not the target for our study. We mainly wanted to inhibit S6K since we don't see any changes in AKT signaling. We have not checked for a specific AKT inhibitor because I think we believe that the effect which we are seeing here is mostly through S6K inhibition, but it would be good to compare a dual inhibitor versus a single target inhibitor to confirm the hypothesis.
Hi, this question is for you, Funda. With the FGFR2 inhibitors moving further up in line, is there any clinical or biologic rationale to be combining them with immunotherapy?
Yeah, there's quite a bit of preclinical data, a lot less clinical data. I think there's a lot of excitement in the space, and there are trials in design as well as, we need more data, however, to really demonstrate whether FGFR inhibition makes cold tumors hot is the, let's just say simplest thing people like to say. The question really is where that would ultimately fit into the clinical algorithm. I think in spite of, as Tim pointed out earlier today, GemCis plus IO didn't really dramatically transform outcomes. It did improve survival. So that triplet combination currently, I think, is definitely in the first line setting. We're now trying to figure out if we had an FGFR inhibitor in the first line, would there be IO combination rationale, or are we going to be looking second line? If so, patients will have seen IO already, and whether adding FGFR inhibitor in that setting would genuinely sensitize a patient who's already progressed on immunotherapy. So these are definitely interesting clinical questions that are difficult to preclinically model.
So there's no evidence, like with the IDH1, that they're less responsive to immunotherapy? Like, is that subtype responsive to immunotherapy?
Yeah, I think we still a little bit need more data because a lot of those studies have been done in with subtype analysis subsequently, and we can't say that those patients are resistant. And in general, that is a little bit better behaving population as well.
If I can just add to my answer to your question regarding the PDX. So as I mentioned, the IDH1 mutant PDX was derived from a metastasis, and the patient was previously treated with AG120 and became unresponsive. So, and our data indicates that AG120 actually makes the cells resistant to dasatinib. So that might be one more reason, and we don't actually know the phospho-S6 levels because we've seen in the dasatinib resistant clones. So we believe that along with the IDH1 mutant status, the phospho-S6 levels is also very important to judge how well the dasatinib or the combination might work. And I think as we get more PDXs, maybe untreated or not AG120 treated, we may see different responses to dasatinib and the combination.
So Michael, I know that you've generated all these FGFR fusions in the context of syngeneic models, so you're probably already systematically looking at the immune effects.
Yeah, exactly.
Do you want to expand on that?
So maybe I might add on that. So to my knowledge, there is, because there has not been a good preclinical model to study that, because people have used human transcripts, and therefore you have to use a host mouse that is immunocompromised. So one of the goals what we're trying to do is to actually address that. We haven't done it yet, but of course that's the goal.
Okay, other questions? If not, can we get time? Can we have the break now? Okay, back at 4 o'clock.
Thanks for the speakers and all the great questions.
Nice to meet you.
Nice to meet you finally.
Yeah,
happy
thank
to
you for the
contribute.
samples, for the study.
Last second thing.
Hi, my name is Kirsten Ernie, and I'm from Chicago. I am a member of Team CCF. I volunteer in order to raise awareness and funds for the Cholangiocarcinoma Foundation in honor of my beautiful mom, Mary.
I got involved with the Cholangio Connect program because when you are first diagnosed, you want to reach out to see if there's someone who's on a like journey, such as yourself.
I decided to become a mentor. It was a really easy decision. My life fundamentally changed when I had heard the diagnosis for my dad, but the foundation provided me with friendship, community, and support. I felt loved and welcomed, and it was clear to me that it was time for me to give back in the same way.
I had some reservations about becoming a mentor. I thought I had to know all of the answers to help other people, and I met an incredible mentee that I now mentor, and I became really close to her. So again, together, that's how we get through it.
And I feel like having a mentor and mentee relationship for a rare disease like this is extremely important because it does help normalize the journey that you're going through, which is very challenging, and also because it helps you understand you're not alone in this.
No one ever deserves to feel alone in this. And hearing that diagnosis, you know, as the patient or a family member, it can be extremely overwhelming. So that is why finding CCF, you know, love, support, hope, that's why we're here.
Hello, I'm Dave Fleischer from Odenton, Maryland. I volunteer for the Cholangiocarcinoma Foundation in support of what I call my H4: hope, help, heal, and honor.
As a patient myself, I'd like to share my appreciation with the Cholangio Connect mentors. Thank you, thank you, thank you for your time, your knowledge, your support, your empathy, and everything that you have given to all the cholangio mentees over all the years that it's been available. As a patient, there are days we are tired, and we need you more by our side than you will ever know. And this is a harsh cancer, and it cannot be easy on you as well. So please be mindful of your needs and practice self-care, because we need you. We need you almost every day. So thank you.
Hi, my name is Linda Fleischer, and I'm from Odenton, Maryland. And I volunteer as a Cholangio Connect caregiver for two reasons. One, to honor the memory of my daughter, Sarah Bennett, and also to support other caregivers walking the caregiving journey, and also for bereaved caregivers who have lost a loved one. Thank you.
The only way to get cholangio known is to get it out there. People need to know what cholangiocarcinoma is. They need to know how serious it is and how we need attention too.
It's been incredible, the outpouring of support from many, many families. I think to date, we are just over $12,500 raised. But I think more importantly to us is the over 90 families that have donated to cholangiocarcinoma, and hopefully help us all raise more awareness around this disease so that we can help those coming behind us have better outcomes. You know, I think for those trying to decide if they should do a fundraiser, I think follow your heart, lean in, think of your loved one and what would they want. We can't change the outcomes, but we can change the future. And there's a lot of momentum with cholangiocarcinoma and what the foundation has done. And I think that more of us can do things. It doesn't have to be big things. Small things help as well. Just telling people and raising the awareness can help those that come behind us.
We all live on the one basis of hope. Hope for a better tomorrow, hope for the next patient to get a better opportunity. And the only way that we can do that is to put ourselves out there, to let our story be known, to make other people want to get involved, want to know what this is, and want to make a difference in the world.
Hi, my name is Cindy and I live in South Fort Myers, Florida. I've been working as an annual conference volunteer since 2018. My brother was diagnosed in 2013 and he was a patient mentor through Cholangio Connect. I have also been involved in a patient caregiver role through this organization's support groups and on social media. My brother and I both found the volunteer roles to be very rewarding, learning much information and tapping into numerous resources, but most importantly, helping to guide patients along the way. We all had different skill sets and talents to offer others. I've found this role to be one of the most important and rewarding parts of my life. I would encourage anyone to get involved and become a volunteer to help along the way. Thanks so much, Cindy.